The main things that we're going to be talking about today are the immunobiology of both PBMC and hematopoietic stem cell humanized platforms. And then we'll talk about applying each one of those different platforms as it relates to preclinical drug lead selection, safety, tox, etc. So as we usually do, let's get started quickly by giving you a reminder of the Jackson Laboratory in terms of who we are, what we do, and what our mission is. Our mission is to discover the precise genomic solutions for disease and empower the global biomedical community in the shared quest to improve human health. Now, we do that in multiple ways. And as a reminder, we are not just a mouse house. We're more than just a repository of strains of mice. We conduct our own grant-funded research programs. We provide education programs based on our science and working with mice. And we work with a number of commercial entities around the world in their drug discovery pipelines because we provide services around these different platforms, as you'll see today. And all this together allows us to develop new innovations and provide things that can accelerate people's research programs around the world. And we do consider ourselves to have global influence. Yes, we started back in Bar Harbor in Maine in 1929, and then later expanded out to the West Coast in California. Out in California, is a mouse distribution center, but it's also where we conduct contract research organization activities. More recently, we've expanded to China. And even more recently, this fall, we're very excited to let everyone know that we've made some purchases in Japan, and we're further expanding our operations and facilities in Japan as well. So to get started today, I've broken my talk down into 4 discrete chapters. So I can introduce to you the different topics, help you understand the immunobiology, and then go into their application. In chapter 1, we're going to be talking about what we commonly call the GvHD platform. However, the better way to think about these sets of platforms is that they are used to interrogate immune modulation of human T-cell responses. So they can be used in the context of GvHD, but thinking about them more broadly in terms of T-cell responses is more appropriate. We're going to use these PBMC engraftment platforms to talk about something called cytokine release syndrome, so we can look at it as a measure of safety and tox of some of these immunomodulatory things, and then show you how you can use it in your drug discovery pipeline to look at the balance between preclinical efficacy and toxicity. Then we'll move into the more sophisticated, what we call hematopoietic stem cell humanized platforms, talk to you about the difference between those different hosts, and then, as we did in the earlier chapter 2 and chapter 4, we'll go in and we'll talk about applying those models to monoclonal antibody therapy, bispecifics, CARs, etc. So before I get started, I'd like to find out a little bit more about our audience and ask a poll question. Check all that apply to you. Are you interested in therapeutic antibody development that is focused on lead selection based on pharmacokinetics and safety? Or not just restricted to antibody, but if you're also developing bispecifics, chimeric antigen receptor technologies, or even small molecule technologies, feel free to check that. But we're also interested in whether you're interested in looking at pharmacokinetics and safety. Therapeutic antibody development focused on lead selection based on mode of action and efficacy, or just strictly safety and tox assessment, and or development of some of these other therapies more specifically. So check all of those that apply to you, and that'll help us get some nice feedback on our audience so that we can tailor future presentations more appropriately. Whenever you're ready, it looks like things have slowed down. Xing, go ahead and display those results. All right, excellent. Thank you very much. Appreciate everyone's participation. There we go. Yeah, we've got kind of a wide spread of interest by everybody. Excellent. That's great. Okay, so chapter 1. Well, let's introduce the PBMC platform and look at some of our abilities to modulate T cells in those animals. Now, to get started, I'm going to point you to this really very nice presentation that covers a lot of the aspects of transplanting PBMCs into the NSG host mice. There's a number of different things that influence the rate of engraftment, the development of graft versus host disease, the mean survival time, weight loss, all these other different factors. And the first of which is whether or not you include irradiation. You can get engraftment even if you don't irradiate these mice. And what the purpose of irradiation is it's twofold. First of all, it's to diminish the host immune cell populations, which gives a competitive advantage to the donor cell populations. But during the ablation of those cells, it can set up a more pro-inflammatory environment. So what this does is accelerates the ability of these cells to engraft and expand in these models. And the ramifications of that are this quicker expansion and reaction against the mouse tissues, which leads to a shortened lifespan, which can also be correlated very tightly with a weight loss. And there's a number of other clinical outward signs of the animals in terms of health. If you leave out irradiation, you're going to slow down this process, but you're still going to get engraftment, and you're going to have a delayed situation of weight loss. Now, what's also important to recognize here is that the dosage of the cells can also make an important influence on the mean survival time of these mice. And as you would expect, the more cells you put in, the shortened survival time because you've increased the rate of engraftment and you've accelerated the development of graft versus host disease. Now, what was really nice and pointed out by this paper as well is that if you knock out just mouse class 1 or mouse MHC class 2, excuse me, that demonstrated very clearly that mouse MHC 1 and 2 are intimately involved with the development of graft versus host disease in this model. So what are the cell populations that are engrafting in these mice? This is looking at 4 different donors, A, B, C, and D. We're looking at flow cytometry of peripheral blood, and we're looking at human cell populations specifically. We're gating out the mouse populations, and we're looking at the expansion of the human based on total CD45 cells at day 5 versus day 10 in all of these different donors. And then you can ask the question using cellular markers, what percentage of these human cells are T cells, B cells, myeloid cells, and natural killer cells? And what you can see is that the T cells expand dramatically over time. B cells are very, very low and are maintained at least out to 10 days. The model does not support myeloid cells well, and the natural killer cells disappear very quickly from the peripheral blood over the course of the first 10 days. So this is primarily a T cell model in all of these different hosts. Now, there's one other aspect to this that's very important to understand, and that is that the rate and severity of GvHD is donor dependent. And what that means is, as you can guess, the human population is genetically diverse. It's trained on different molecules that it's exposed to in the thymus. So the responses are going to be different. So when they see these class 1 and class 2 molecules in the recipient, their strength of response to those mouse MHC molecules is going to be different. So every PBMC donor is going to, the engraftment kinetics of every PBMC donor is going to be slightly different. However, it is highly reproducible. So once you have a collection of cells, you can understand the kinetics of those cell populations and reproducibly recreate that each time that you engraft those numbers of cells. So let's take a quick look at an application for these mice that was pulled from the literature. Now, in the human population, the most common use of bone marrow transplantation is to treat different forms of leukemias. Now, what you really would like to see out of the bone marrow transplantation is a beautiful graft versus leukemia response to clear the leukemia out. But sometimes, and usually greater than 50% of the patients are going to develop either acute or chronic graft versus host disease, unfortunately. So individuals, clinical Researchers and physicians would like to be able to maintain that graft versus leukemia response, but be able to manage GvHD in the recipients. So this group of scientists took very close look at the human population to try to identify markers and cell types that correlated with the development of graft versus host disease. In human patients that had this most severe graft versus host disease had significant involvement with the gastrointestinal tract. So they took a close look at those cell populations through a number of different techniques. And what they ultimately found was that there was a population of plasmacytoid dendritic cells that had upregulated ICOS ligand expression on those cell surface. And coinciding with that was a T cell population that was also ICOS positive, and they were based on cell surface markers and transcriptome, they were characterized as Th17 T cells. So the combination of these two things were very intimately associated with a GI-associated graft versus host disease. Now, to prove that this was the case further, they went back to the mouse studies. And what they did here is they first isolated human PBMCs and completely depleted them of all plasmacytoid dendritic cells. They isolated the PDCs and put them in culture, and they stimulated them with CpG, which stimulates through the toll-like 9 receptors. And they also added FLT3 ligand. This stimulates the development and differentiation of dendritic cells. But in combination, this created a very concentrated population of ICOS ligand positive plasmacytoid dendritic cells. So they created 2 study cohorts, each injected with these dendritic cell depleted PBMCs. Half of them got these expanded DCs, and half of them did not as the controls. And when the mice received these expanded plasmacytoid cells, that increased clinical scores indicating that they were developing GvHD. This would be hunch posture, ruffled skin, with lethargy, cold to the touch, those kinds of things. But also accelerated the mice needing to be euthanized based on the development of graft versus host disease. So demonstrating that this was the cell population and looking at the things that these different cells expressed on their surface, which was involved with activation, they wanted to test a molecule that they had developed to try to suppress this pathway. Now, they used a control molecule called belatacept, which is clinically approved for organ transplant prophylaxis. And the way this molecule works is it's a fusion protein that binds CD80 and CD86 on the antigen-presenting cell and prevents the molecule from binding to either CTLA4 or CD28. Most importantly is the protection from binding from CD28, because that's the co-stimulatory molecule of the T cell in the context of TCR engagement that activates the T cell, and it blocks activation of these T cells. Now, these researchers, recognizing that ICOS was a very important co-stimulatory pathway as well, they decided to develop a molecule called a bispecific monoclonal antibody, because it has 2 different specificities. One arm binds to CD28 and prevents its binding to CD80 and CD26, and the other arm binds to the ICOS and prevents that pathway. So this single molecule can bind both of these, preventing the co-stimulatory activation of the T cell. Now, also very importantly is this end of the molecule called the Fc arm is also highly engineered. This is important because what is being done here is that the Fc portion is very important for binding to neonatal Fc receptors, which is important for maintaining half-life activity and increasing bioavailability of the molecule. But also importantly, they've excluded binding to Fc receptors and C1q, which is involved with complement binding, because that can lead to different toxicities and different off-target effects that they wanted to avoid. And we'll come back to some of these safety things with regard to cytokine release later in the story. But first of all, I want to bring your attention to their dosing of these mice. So these are human PBMC engrafted mice, and then they were dosed with this ALPN 101 versus belatacept. They used different concentrations of the drug that were dosed 3 weeks for 3 times a week for 4 weeks. The Batacept was a higher overall dosage, but the same dosing regimen. And then they tried the test compound at 100 micrograms single dose on day 0. So this is a prophylactic dosing protocol. So if I concentrate your attention on the blue and the brown here, which would be the belatacept control versus the single dose of their test article, we can see that there was a very similar reduction in disease activity index, a very similar maintenance of body weight change, and a very similar overall survival. But if we take a look at even the low dose given 3 times a week for 4 weeks at 20 micrograms, we saw even further reduction of disease activity index, even better maintenance of body weight, and a long-term maintenance of 100% survival throughout this study, indicating that this drug had a very good response in the system. But this also points out that you can use a number of different measures in this in vivo system to really understand overall what's happening both mechanistically and health-wise for the course of these animals. So in summary, the human PBMC engrafted mice are a model of T-cell mediated GvHD. The engrafted human T cells can be used to evaluate preclinical immunomodulatory therapies. And you could do this either for anti-inflammatories, which we just demonstrated, but you can turn this around and turn it on its head, and you can actually use it for pro-inflammatory testing molecules, where you want to stimulate activity of T cells. In that case, you're looking for accelerated development of GvHD. So there's a lot of different readouts that we didn't mention so far. You can use flow cytometry and immunophenotyping. We talked about disease activity, body weight, and survival, but you can also include tissue pathology. And as we'll see in a few minutes, quantitative human cytokine and chemokine expression to really tell you more about your mechanisms of action and safety. So in chapter 2, let's talk about some more very discrete applications of this platform. And what I want you to understand is this is an extremely modular platform. And we'll show you why that's the case as we go through the subsequent slides. But partly is you can either include tumors or not include tumors. You can put tumors on board first and then put in the PBMCs, put in PBMCs first and then put tumors on board. It depends on what tumor you're working with and the scientific question that you're asking about. And so therefore, you can use this in multiple different ways to tell you about your lead selection pipeline and do preclinical discovery. You can use single donor assays for lead selection, using the same donor to compare different leads side by side. Or if you get more advanced and you really want to understand donor to donor differences in PBMC responses, you can design these studies that have multiple different donors so you can get an idea of what kind of experience you might get to in those stage 1 clinical trials. So let's talk a little bit about safety and efficacy. And before I run into the data, let me start with a little bit of a backstory on this. So many of you may be familiar with this story, but I'll run through it as a reminder anyway. So back in 2006, there was a drug called TGN-1412. This was a drug that was going to be moved into the clinic. It had gone through a number of assays, both in vitro and in non-human primates, to check its safety in terms of not developing a pro-inflammatory cytokine release syndrome that would cause organ and tissue damage. So it was negative in the in vitro cytokine release assays that it was run through, and it was negative in these non-human primates. So it moved into human patients and unfortunately caused very, very severe toxicity. And there's been a number of molecules over the years that have developed this in the clinic. And the ultimate situation is that it was recognized that these assays that were in place were not adequate to really measure the true safety of these drugs. So our innovation team here at the Jackson Laboratory, led by Dr. James Keck, set out to develop a better in vivo assay that includes human cells. And the importance of this is to really look for a systemic response. Now, to give a little bit more backstory, the TGN-1412 binds to the co-stimulatory molecule CD28. But what it can do is the nature of its binding is such that it can form complexes and can activate T cells even in the absence of TCR engagement, which is different than a typical anti-CD28 monoclonal antibody that binds to CD28 but needs engagement of the TCR to get stimulation of those T cells. So what Dr. Keck did is he took the NSG mice very early on, only about 6 days after engraftment of the PBMCs, treated with them with anti-CD28 or TGN-1412, and measured human cytokine release, interferon gamma, TNF-alpha, and IL-10. Looked at this response across 3 different donors and saw a strong cytokine release response. Okay? So what's important here too is we're seeing a cytokine release from known agents that cause this type of cytokine release, and there are donor-to-donor differences in response, similar to what is observed in the clinic. Now, what's also very important about this is if you compare these types of activities for an in vitro assay, you can get a release of cytokines, but what does that really mean? In other words, is the spike in response going to lead to systemic toxicity? Or is it the duration of high cytokine release? Or is it just a quick spike of response that the host is going to recover from very quickly? It doesn't tell you anything about how the organism or the recipient of these treatments is going to respond overall. So the value of doing this in vivo is that you can use things like clinical scores and body weight, et cetera, to tell you what these responses are. So if you look at the previous slide, the cytokine response was higher for CD28 in this particular dosage that was used here than the TGN-1412. And because of that higher cytokine response, we had earlier increased clinical scores and an earlier loss of body weight than one with a lower cytokine releasing with the TGN-1412. So you can get a systemic measure of these responses. Now, the other interesting thing about this is that this is very highly repeatable. You can get the same types of responses as long as you use the same PBMC donor and the same types of dosages over and over again. So you can go back in and actually use these molecules like anti-CD28 or OKT3. OKT3 is an anti-CD3 monoclonal antibody, stimulates a very strong cytokine release response. And you could use those as positive controls to stimulate this response across different donors. And again, you'll see different magnitudes of response with different donors. And then what you could do is you can also ask questions about, well, can we tamp down that response? So here we're in the other arm of this study, we're treating these mice with the anti-TNF-alpha monoclonal antibody Humira. So we very dramatically knock down TNF-alpha, but then we can also ask the question, does this also lead to reduction of interferon gamma and IL-10 and other cytokines that are part of these broadly expressed pro-inflammatory platforms? And the answer is yes. So you can use this platform in either direction. So it's very modular. Now, we talked about one form of bispecific antibody, but there's also other forms called bispecific T-cell engagers. And the construction of these are a little bit different. In this particular instance, we've got the binding portion of one monoclonal antibody linked to the binding portion of another antibody. Now, this one end is specific for CD3 on T cells, and the other end is for tumor-associated antigens. And the goal here is to use this molecule to bring this T cell into context with the target cell population that you wish to destroy, and do this by bypassing the normal TCR receptors on the surface of the T cell, but get these T cells to be activated and have a cytotoxic response against the target. So in this particular study, we've got the NSG mice, and in this case, the bispecific that's being used here is to the epidermal growth factor receptor on one end and CD3 on the other end. Also included here is the anti-CD28 and Keytruda. Keytruda is the commercially available, clinically approved anti-PD-1 checkpoint inhibitor not particularly known for cytokine release. So it's really kind of a negative control for cytokine release. But we do know that bispecific T cell engagers are well known for inducing cytokine release. So half the mice did not include a tumor, and the other half did. And the tumor that was used here is a luciferase tagged single cell suspension introduced IV to mimic leukemia responses, etc. And you can image the mice for the tumor by luciferase imaging. Now, one of the things that's important here is that in the absence of injected tumor, just the presence of human PBMCs and inclusion of these bispecific molecules, you do not see a cytokine release response. It's only in the presence of the tumor that you see this high cytokine release response. Now, the inclusion of Keytruda is to block that checkpoint inhibitor pathway to see if this would further enhance cytokine release and see if it would also enhance clearance of the tumor by further accelerating these T cell responses against that tumor. And in this case, this tumor is EGFR positive. So we didn't really see an enhanced clearance of the tumor by including Keytruda. That was actually quite similar to the EGFR-CD3 bispecific alone. Keytruda by itself did not enhance clearance of the tumor, but very interestingly, anti-CD28, which is known to have activation-induced depletion and cell death activities, led to a T cell cytopenia, and which was believed to allow outgrowth of that tumor, and the tumor actually expanded in those mice. Now, if we look at the overall clinical scores in body weight in this treatment paradigm, we can see that the inclusion of Keytruda co-treatment really led to an earlier and longer duration of negative clinical score. So the mice were actually a little bit unhealthier overall, but they did bounce back and seem to recover. And so there's also the cytokine release increased clinical scores with the bispecific alone. And this was also reflected in lowering the body weight, also indicating that these mice were a little bit ill, but they did bounce back and recover. What's also interesting here is, remember, there was no massive cytokine release in this particular study with this dosage of anti-CD28. The T cells were somewhat depleted, and this led to the outgrowth of the tumor, and this led to a tumor burden-induced increase in clinical scores and a late induction of loss of body weight or poor health in these mice. So you can tell a lot about what's going on in your study by doing these very important in vivo studies. Now, the other important question here is, which we get questions about, and that is, well, what is the influence of GvHD in the context of these tumor efficacy studies when we're modulating these T cells to respond against the tumor? The studies that I've shown you, most of which so far are mostly done prior to the onset of GvHD, which is usually after about 10 days or so. However, it is important to us to develop a model that would be devoid of these overlapping types of activities. So based on that King paper that I showed you earlier, I mentioned that there were class 1 and class 2 individual NSG mice showing the involvement of these molecules. So Dr. Lenny Schultz and Dr. Michael Brem took this a step farther, and they created what we call an MHC class 1, class 2 double knockout. There's actually 3 components of the genome that are knocked out, but we call it the MHC1/2 double knockout because it's knocking out both class 1 and class 2. So how does this compare in terms of engraftment, survivability, body weight loss, etc.? So first, we're just looking at the engrafted cell populations shown here. We're comparing the double knockout to the NSG mice, and this is just 5 days after engraftment. We're seeing massive expansion of the T cells, and just like before, very few B cells, very few monocytes, very low numbers of NK cells, and also very poor support of CD33 positive myeloid cells. What we see after 10 days, as I mentioned a moment ago, we start to see body weight loss and loss of survival of those NSG mice. But if we look at the double knockout, we can see that the mice are very healthy long-term, maintain long-term survival. Now, it's important to note that the cell population that's injected and the concentration and numbers of those cells that are injected can influence these kinetics. You can give too many cells and end up, even with the double knockout mouse, causing this type of profile. So every peripheral blood mononuclear cell population does need to be titrated very carefully. But it does allow you to do longer term studies without seeing this overlapping problem with GvHD responses, and allows you to do longer term tumor studies. And I'll show you more evidence for that in just a moment. Now, I talked earlier about inclusion of irradiation or exclusion of irradiation. The prior data set that I just showed you included irradiation, but you can also leave out radiation in this model and get engraftment. So here's comparing those two again. In the absence of radiation, you get lower levels of engraftment initially, but by the later stages, they begin to more or less equalize once you get out to 10, 11, 12 weeks. Again, the GvH that's going to be expanding in the NSG is going to lead to a shortened survival, but you definitely have much longer survival with a double knockout. And so you've got a full 75 or 80 days, depending on the PBMC donor, in which to work with those mice. So the other question is, well, in the absence of GvHD, can you use the double knockout to see cytokine release responses? And the answer is yes. So here's a comparison between the two. We've got Keytruda as the non-CRS control. Also Yervoy, which is ipilimumab, which is an anti-CTLA-4 monoclonal antibody. That's not particularly known for cytokine release, but the OKT3 anti-CD3 monoclonal antibody is known for very strong cytokine release. And so with this particular donor, the responses across the board were very similar between these two models. And this has really become the model of choice for us when we do CRS assays. So I mentioned that this model would allow you to do longer term studies and increases the modularity of what we can do. In other words, now what you can do is you can do solid tumor studies. So what you need to do in that context is take this class 1, class 2 double knockout, take solid tumors and put them on board first. This is a breast tumor CDX model that was engrafted into the mammary tissue. When those mice are ready, which means that their tumors are uniformly at about 200 millimeters cubed, roughly. 5 days before dosing, you can introduce your PBMCs in the absence of radiation. And the reason you want to leave out irradiation is because you don't want to irradiate your tumors, because that could influence the tumor growth of those tumors. Because we only want to look at response following our drug treatment strategy. So 5 days after PBMCs, at day 0, we begin the dosing. And in this case, we're using the EGFR bispecific, and we're comparing that to cetuximab. Cetuximab is also specific for EGFR, and it's a monoclonal antibody. There was very poor response with cetuximab, but a really nice tumor response with the EGFR bispecific. This is the average data. This is the individual mice. And we looked at body weight change, indicating that out to 40 days, these mice maintain body weight and were very healthy, and demonstrating that you can do these more longer-term studies. So JAX has developed a new and reliable in vivo cytokine release assay to evaluate new T-cell agonist immunomodulatory therapeutics for cytokine toxicity. The in vivo cytokine release assay can also be used to evaluate anti-inflammatory therapies. The new assay is highly modular and can include tumor targets necessary for evaluation things like bispecific T-cell engagers. The in vivo assay provides a systemic analysis of the consequences of cytokine release that the in vitro assays simply cannot measure. And the new NSG class 1, class 2 double knockout host supports human PBMC engraftment, allows analysis of therapeutics for CRS, and it allows extended tumor preclinical efficacy studies in the absence of acute GvHD. So now let's go on and introduce to you the CD34 engrafted models. So for those of you who are not completely familiar with hematopoiesis, in these models, what we're doing is we're transplanting human cord blood-derived hematopoietic stem cell populations that engraft into the bone marrow space of the mice, and they differentiate out into these multi-lineages that move out into the peripheral blood and peripheral lymphoid organs. And so this is what we call multi-lineage engraftment. Now, the NSG host is the gold standard for this. It does have multi-lineage engraftment. However, certain cell populations like natural killer cells and many of the myeloid populations are there, but they're in low frequency. And that's because many of these human cell populations really require human-specific growth factors for their support and differentiation. Therefore, mice like the NSG-SGM3, which expresses human IL-3, CSF2 or GM-CSF, and human stem cell factor or KIT ligand, improves both myeloid development and, as you'll see in a moment, regulatory T cell development. The IL-15 mice was created. It expresses physiological levels of human IL-15, and this improves natural killer cell development. So if we just do a side-by-side comparison and look at these subpopulations of cells based on these cell surface markers by flow cytometry, we look at 12 weeks post-engraftment, we can see that in the SGM3 we've increased overall engraftment, CD4 population. Within that CD4 population is the regulatory T cells compared to the NSG, and have expanded the myeloid population. We do a similar kind of study looking at a snapshot 12 weeks post-engraftment comparing NSG to the IL-15. Most of these cell populations are very much the same, but we have a significantly increased percentage or frequency of CD56 positive, CD16 positive human natural killer cells. Now, there's also engraftment over time, and so now I'm just showing you a comparison of the NSG to the SGM3. And I'm really just broken it down to looking at the Tregs and the myeloid cells, which are the two populations that are most significantly expanded in these models. And you can see that the engraftment in the NSG is low but stable over time, and the engraftment is much higher, but then kind of tamps down over time. And one of the questions that we get is, well, why is there an early spike in these myeloid populations that comes down? You need to remember that this is frequency. This is the percentage of these myeloid cells out of the human CD485s over time. So as the T cell populations expand as a frequency, the concomitant drop in the frequency of the myeloid cells is expected. And so these things do change over time to give you an idea of the growth kinetics of these different cell populations and give you an idea of when and how to and where to work with these mice over time. So in the IL-15 mice, there's been quite a bit more activity in terms of characterizing these cells. These human NK cells isolated from the spleen of these mice express all of the up and down regulatory cell surface molecules you would expect to see in human NK cells. Their development in terms of human granzyme B expression is more developed compared to the NSG. There's a subpopulation that is granzyme B low. If you take these mice, the NK cells from the IL-15, and put them in a killing assay, they have very comparative killing assay to NK cells isolated directly from human peripheral blood. So to summarize this very quickly, JAX creates and distributes these mice that are engrafted with human cord blood-derived CD34 hematopoietic stem cells. So to be clear, we're engrafting these mice on a monthly basis, all three of these different host strains. Therefore, you can go to the Jackson laboratory and buy these verified engrafted mice off the shelf. Many people decide to set up these programs in-house, and I think ultimately they discover that there's a lot of work that goes into that, and the cost of doing that is probably more than simply buying these mice off the shelf and rolling them directly into their drug discovery program. These mice develop and maintain stable multilineage immune cell engraftment. Certain human immune cell populations require human-specific growth factors for the development and function, and that's why you need to choose your model carefully. So the selection of the platform will depend on the cell population and your immune mechanism of interest. So in the last chapter today, let's run through some of the applications and uses of these different platforms. So one of the first things you need to know is what happens when you put a tumor on board these mice. And we often get asked the question, well, if you've got a fully developed immune system in these mice, why can you get tumors to engraft on these mice? Shouldn't that immune system simply reject these tumors? And that's what I first thought, being an immunologist, that was my presumption as well. Well, there's a number of different mechanisms that allow these tumors to grow. Some of which is the fact that yes, there are components of the immune system that are not fully functional, like B cell responses, antibody production, and a few other things. But there's also downregulation of class 1 and class 2 molecules on these tumors. There's upregulation of checkpoint inhibitor pathways, and there's a wide range of other things going on, some of which lead to really no changes in the growth kinetics of these tumors over time. But some of these tumors indeed are rejected and really don't grow well on these humanized mice at all. And another part of this is the immune cell infiltration of these populations. Just like what is observed in the clinic, some of these tumors are very highly infiltrated with human immune cells. Some are what we call cold or have very little immune cell infiltration. So if you have a favorite tumor that has never been put on board a humanized mouse, one of the first things you need to do is a pilot study and just understand whether that tumor will grow in these mice and what the growth kinetics are on those mice with and without the presence of the human engraftment. Now, this paper is very nice in the sense that it also took a deep look at, well, what is influencing the entry of, in this case, in these particular graphs, the entry of human T cells into these tumors. The overall conclusion here is that the T cell infiltration frequency is driven by the tumor and not the donor. So what they did is they looked at multiple different donors across multiple different studies, and each one of these different tumor types was controlling the types, frequency, and levels of T cells that were entering into those tumors. And this makes sense because these cell populations are expressing chemokines and other different things, have different receptors, so they're going to have different chemoattractants for these different cell populations. The other thing to mention is that some tumors are very myeloid infiltrated and some are very T cell infiltrated. So the other part of your pilot studies to really understand what you're working with and mechanism of action is during those pilot studies where you're looking at growth kinetics is to harvest those tumors and do deep phenotyping of those tumors to understand what's going on. So based on all these different things that are going on, what kind of immune modulatory things can you do? Here's an example of taking regular NSG mice that are CD34 humanized and then putting a tumor on board. So this is a solid tumor injected into the mammary gland, and then treated with either pembrolizumab or nivolumab, which are anti-PD-1 checkpoint inhibitors. And you can see suppression of tumor growth with two different donors here. Now, what I want to point out here that's very important is there are donor-to-donor differences in response. We see that checkpoint inhibitors in particularly, we see about a 30 to 35% response rate. And this is very similar to what is observed in the clinic. And we do have clear examples where we put the same tumor, but a different donor, treat with these drugs and see absolutely no response. So it is a challenge, but it is something that you need to account for this by trying to include multiple donors in both arms of your treatment studies. So you can understand whether this a lack of response is due to a study failure or it's specifically due to the particular donor that you're working with. Other approaches include cell depletion. Here's a nice paper where they developed a monoclonal antibody against GITR. GITR is highly expressed on regulatory T cells. Regulatory T cells are within the tumor, can lead to suppression of CD8 T cell responses. The Fc portion of this anti-GITR monoclonal antibody is able to engage with NK cells or myeloid cells to initiate antibody-dependent cellular cytotoxicity or cellular phagocytosis, which can lead to depletion of the cells, which tips the scales and allows those CD8-mediated responses to go forward against the tumor. You can see suppression of tumor growth. You can harvest what's remaining of those tumors, interrogate them for depletion of the Tregs, and in fact see reduction of Tregs in the tumor. And you can also look for cytokine responses. If you have increased activity of your CD8 T cells, you should expect and do see increased IL-2, increased interferon gamma release. If we take a look at the NSG-IL-15, and we take those humanized mice and we engraft them with a NK cell resistant Dowdy tumor cell. This is a B cell lymphoma that expresses CD20. And we treat them with rituximab, which is a clinically approved monoclonal antibody, which is anti-CD20. We can see reduction of tumor volume in those mice, indicating some ADC-mediated killing of those tumors. What about bispecifics? Now what we're doing is taking humanized NSG, again putting these tumors on board through the mammary cell engraftment, and then treating these mice with the EGFR-CD3 bispecific to get those T cells that naturally form from those hematopoietic stem cells to respond against those tumors. And we did this study as a prophylactic trial and a therapeutic trial. And what I mean by that is, in the prophylactic trial, we started with the mice enrolled into the study with very small tumors, versus a therapeutic trial, meaning the tumors were twice the size, so higher tumor burden. And in both of these cases, we saw very, very nice reduction of the tumors using this bispecific T cell engager. This is the average data, and this is the individual mice shown here. So very nice responses. Very early on, you can do a number of other measures, like look at cytokine release. And we can see that with some of these cytokines, tumor burden is associated with even very much larger. Look at the scale of the cytokine release of interferon gamma and IL-10. Tumor burden typically in the clinic leads to more higher cytokine release responses, more significant changes in activity of T cells. You can use flow cytometry to look at mechanism of action. There's a lot of different readouts that you can use in this platform. Shown here is a very interesting study. Something we haven't talked about yet is chimeric antigen receptor therapy that many of you may be familiar with. This is where you take a patient's own T cells and put them in an ex vivo environment, use viral delivery of constructs, which create a binding receptor on the exterior portion of the surface and an activation arm in the cytoplasmic arm of that construct that's expressed in the cell membrane. This is a clinically proven approach for B-cell lymphomas. There are CD19 CAR Ts that are available clinically. What these researchers were wanting to do was expand this beyond CD19 B-cell lymphomas and move into what's called a CD44 positive monocytes, which also express CD14 on their surface. And what they did in this study is they used the CD34 engrafted SGM3 mice. Because they have higher production of human T cells in them, they isolated the T cells from these mice, put them in culture, transduced them with their two different CAR structs, one for CD44 and one for CD19, and used them to look at B-cell or monocyte aplasia, clinical signs of cytotoxicity, and different cytotoxicity markers. So the first question was, once they've created these CAR T cells and CAR M cells, just put them back into SGM3 CD34 engrafted mice. And what's nice about using these as donor cells is that these T cells are trained against the mouse thymus. So when they go back into these mice, they see the mouse cell's tissues as self, and there's no graft versus host response. So in blue is the CD19 specific CAR T, and it depletes these bone marrow derived CD19 B cells. And the CD44 CAR T depletes CD14 positive monocytes and clears them out quite nicely. They also looked at cytokine release during the depletion of these studies, and they saw changes in body weight with both of these, increases in cytokine signaling, changes in body temperature that's also associated with cytokine release. And they also looked at organ and tissue damage. And one of the markers for liver damage is increased serum amyloid A, indicating that there was some liver toxicity in this model. So you can use this type of platform for look at some of those cytotoxic activities. But they wanted to take this a step farther. And what they did demonstrate is that if they did put B-cell lymphomas or myeloid tumors into these mice, they could see depletion of those tumors, they could see cytokine release. And one of the things that they would like to do is ask the question, are there other treatments that we can use that will block the toxicity of the cytokine release, but not inhibit the response against these tumors? So they tried two different treatment strategies. One was tocilizumab, which is an IL-6 receptor antagonist, and anakinra, which is IL-1 receptor antagonist. Neither of these molecules interfered with the clearance of tumors using these two CAR T-cell populations. And then they asked the question, well, what does it do for survivability? And overall, the anakinra improved overall survivability. And then one of the consequences of toxicity beyond liver damage is also neurotoxicity. And one of their markers or measures of reducing neurotoxicity is changes in meningeal thickening. And they found that the anakinra treatment reduced meningeal thickening in this particular model. So there's a wide range of different questions that can be asked with these very sophisticated models. And my very last example today is a CAR-M model. And in this particular case, what they're doing is they're actually genetically modifying the myeloid cell population, not the T cell population. And they use a very similar approach to the CAR T, in that the extracellular domain is specific for a tumor target. In this case, it's for HER2 positive tumors. And the intracellular domain is actually very similar to what is used in CAR T. It's a CD3 zeta intracellular domain, which is part of the CD3 signaling molecule. And it's interesting is that this molecule has homology with the Fc common gamma chain of FcR1. And this can initiate phagocytic response similar to what is found with the FcR1 mediated ADCP responses. So the first thing that they did is they took these genetically modified macrophages and mixed them in culture with SKOV human ovarian tumor cancer cell line that is HER2 positive and look for changes in specific lysis using either untransduced or empty macrophages. So this was really a CAR-mediated increase in phagocytosis. Now, what is also interesting about this is they did some phenotyping of these myeloid cells and found that the adenoviral delivery vehicle also polarized these human macrophages to an M1 pro-inflammatory phenotype. And so this is advantageous because there are myeloid cells that enter the tumor that are of what we call an M2 phenotype, which would be an immunosuppressive phenotype. So taking this beyond liquid culture, they went back to the SGM3 mice. They introduced the tumor, and then they introduced their macrophages that were either untransduced or they had the CAR-mediated transduction of these cells. They introduced these all intraperitoneally, and they looked for suppression of tumor growth. And a large subset of these mice did indeed show tumor suppression and expansion of their overall survival. So showed some preclinical efficacy for this model. So they took it a little bit farther and did something that was very interesting. And they went back to the humanized mice to ask some questions about what were the influence of these myeloid cells when they entered the tumor. And so what they did is they took the CD34 engrafted SGM3 mice, which were engrafted with female human hematopoietic stem cells, and the human myeloid cells that they transduced with their adenoviral vector to introduce their CAR molecule were derived from a male human donor. And that way they could use sex differences to tell a difference between their CAR-introduced myeloid cells and the myeloid cells that developed from the expansion of the hematopoietic stem cells, because both populations would enter the tumor. They used a number of techniques, primary single-cell RNA sequencing, and they realized that these cells that they introduced, the CAR cells, maintained their M1 phenotype in the tumor microenvironment. But also very interestingly, when they looked at the female myeloid-derived cells, those cells were also converted to an M1 phenotype. So the pro-inflammatory responses of these introduced CAR cells also influence the other cells in the tumor microenvironment. So I thought that that was very, very interesting and a very sophisticated, detailed phenotyping of mechanism of action of their molecule. So in summary, the human immune engrafted mice are able to co-engraft either human tumor cell line derived xenografts or patient derived xenografts. We have a number of studies where we've done that kind of work. The tumor immune cell infiltration is controlled by tumor intrinsic factors. The engrafted human immune cell populations are responsive to monoclonal antibody therapeutics that can up or down regulate cellular responses. Human target specific clinically approved experimental monoclonal antibodies can be evaluated for efficacy and mechanism of action. The human immune engrafted mice are highly modular platforms capable of interrogating monoclonal antibodies, bispecific, chimeric antigen receptor for preclinical efficacy, safety, toxicity, etc. So overall, JAX has developed a highly modular PBMC engraftment platform for very rapid preclinical analysis of immune modulators. And they can be used for both pro-inflammatory and anti-inflammatory responses. And we feel that this is a much improved assay for cytokine release responses. It allows you to look at systemic toxicity so you can really understand what do these cytokines that are released actually mean in the context of the whole animal. And JAX is producing a wide range of humanized platforms. They're study ready to roll right into your platforms that you may want to use in-house, or you can come to the Jackson Laboratory, as I mentioned, and we're running contract research activities. We have expertise with these models. You can give us your therapeutic, and we can run those entire studies for you. And we've done this in a wide range of settings across monoclonal antibodies, bispecifics, CARs, et cetera, small molecules, whatever it is that you'd like to do. So JAX is not just a mouse house. We're providing a number of services. We're creating new innovative models all the time, characterizing them, sharing with you what those models can do. So if you have some questions about any of these models, please feel free to contact us.